Great.
Okay. Hi, everyone, and thanks for coming here. We have Bastian Rieck with us today as our speaker. Bastian started his academic journey by studying mathematics at the University of Heidelberg in Germany, where he continued to do his PhD in computer science. He then went on to Switzerland, specifically to ETH Zurich, to do his postdoc and then a senior assistant position with Carsten Borgward in the realm of machine learning. So combining his previous expertise in mathematics and topology, and then combining that with new application areas in machine learning and data science, etc. He's now a PI at the Institute of AI for Health at the Helmholtz Center in Munich in Germany. And he is also has various other positions, such as the co-director of the Applied Algebraic Topology Research Network, which is a very nice name, I think. Bastian has received a number of scholarships and grants throughout his career, and he is also very active in the community in various functions. So he is an outstanding reviewer at many of the leading conferences in his field. So he really cares about improving both methodology, but also, I think, community. That also shows in his blog, which has millions of page views, I've noticed, which is quite impressive, I think, and will be even more impressive if you keep it up during your PI tenure, I think. So, and as a fun fact, Bastian is a Stoic. He's I would say a vehement Stoic, and he's very reflective, I think, in his work, but also in his outreach via his blog, etc. So with that, I hand it over to you, Bastian. Tell us all about your exciting research.
Thank you very much. I should maybe also add that as an additional fun fact, I'm also an ordained minister of the Universal Life Church, and I had the pleasure of marrying my best friend and his wife. So not marrying them in the sense of we now being a couple, but officiating their wedding. That was, I would say, one of the high points of my career so far. But yeah, thank you really for this kind introduction and for this opportunity. So today I'm going to talk about things that are more or less finished at the moment, but that are still continuing. So it's a research direction where we close one branch with a nice set of publications, but we're also opening up some additional branches now, I hope. I have called this talk Zoom and Enhance towards multi-scale representations in the life sciences, with the idea that sometimes it's good to have the proper perspective in life or when you're looking at data. So for instance, when I show you this graph, and maybe the x-axis is already named in a very suggestive manner so that maybe you could guess what is going on here. But so when I show you this graph, everything looks kind of neat, right? And maybe the more mathematically inclined of you would say, oh, well, this is more or less like a constant function, or at least there's not a lot of variance going on. But now when we zoom out, we see that, whoops, something is clearly going on. And by now, of course, you have guessed that this is something on climate change, namely the annual mean temperature. And you can see that we're definitely heading towards a problem. This is not a talk where I could outline what we could do about climate change or how to combat it and whatnot. This is more like showing if you pick the right perspective, you suddenly see patterns in your data that you haven't seen before. Whereas if you zoom in too much, maybe you might miss the forest for the trees and vice versa, of course. So this talk has two parts. The first is going to be about antimicrobial resistance prediction, and the second one, but we'll see whether we reach that in time, because I can go into more details as the audience members might want to interrupt me or want some clarifications. The second one will be about some MRI data analysis using topological methods. So the first one is relatively light on topology, I would say. The second one is well, more dense in that respect, but both are tied together by the idea that it's good to pick multiple views and multiple representations of a data set. So first, some context, and I should stress that as Daniel so kindly said about my biography, I mean, I come from mathematics. I don't come from biology. I'm super interested in these techniques and in the domain, but I also cannot pretend to be an expert, except maybe on Twitter, of course. So there, of course, I'm an expert in everything, like every other Twitter user there. But let me talk a little bit about antimicrobial resistance, why that is problematic, or why that is a problem on its own. You might have even some experience with this. I mean, I hope not for your health's sake, but sometimes when you get an infection, you have to treat it with antimicrobials. And in particular, when you're older, when you have existing comorbidities, this might pose a significant risk for your health. And this gets exacerbated by the fact that some microbes have become resistant to certain antimicrobial products or antimicrobial drugs. So essentially, there is this fear of the super bug, as it's sometimes called, that cannot be treated anymore, and where you essentially have to run the infection and just wait for it to finish. Some well, more or less well-known media organizations have picked up on this. This comes up regularly in the news cycle, most of the time when there's nothing else to say. But now, of course, in the wake of the recent pandemic, which I know is viral based, so it's a little bit different. But even so, some folks have picked that up again, and they said that, okay, it's time to confront the pandemic of antibiotic resistance, because antimicrobial resistance also results in a lot of suffering, a lot of needless suffering, and in a lot of deaths, unfortunately. It's also sometimes called the world's next big health emergency, which apparently is already here. This depends a little bit on where you're living, but it can be a problem in particular in developing countries. And what is certainly also true is that millions are unfortunately dying from these drug resistance infections. So we need to do something about that. We want to find out how to help patients in the end. As a small spoiler, I can say that the methods that I'm presenting here, they are not yet used in the clinic, but my collaborators are preparing some clinical rollouts as we speak, or at least some pilot studies. But of course, getting from the theory or getting from, hey, I have implemented a nice model, to the actual application somewhere is a very tedious and a very long process. It needs a lot of patience, and it needs a lot of patience. So it's not easy to go there, but it's at least it's our goal, or at least the goal of my collaborators. Now, how do we approach this? We do what I think many engineering type people would do. We co-opt an existing technology to solve the problem for us. In this case, we co-opt MALDI-TOF mass spectrometry. So MALDI-TOF means matrix-assisted laser desorption ionization time-of-flight mass spectrometry. This slide contains more or less all the knowledge I have of this technology, because I've never used it in practice. I just get the data and know what to make of it. But from what I understand is that you have a laser source, you have your sample on a mixing plate, or on a matrix as they call it. So it's not one of those mathematical matrices, but it's an actual matrix, one of the first confusing factors for me. You shoot the laser at this sample, and this results in the usual chemical or physical changes of the sample. So you can either have some desorption of energy, you can have ionization, or you can have molecular ions, and then you measure these things with the usual time of flight mass spectrometry. Now, this is nice. This is a nice way, because you can quickly get an overview of the sample composition of the microbiome, and the resulting spectra. I'll show you one on the next slide. They are known to be highly characteristic of a specific microbial species. And this also means that it's now a standard tool for species identification in clinical practice. So when the doctors don't know what you have, like an E. coli or a pneumonia or whatever, they just use that technique to at least identify the species of the microbiome. As we shall see subsequently, this does not mean that they know how to treat it, but at least they get a better overview of what is going on. Now, this is an example of how such a spectrum looks. You have essentially an M/Z ratio, so that's like a mass over charge ratio, and then you have some densities. And the idea is, of course, that you now use these peaks here in order to characterize the data to a certain extent. So far, so good. But the usual antimicrobial treatment workflow looks a little bit like this. You have a sample collection from the patient, so some sputum or some other things that you take from the patient. You wait 12 hours to 24 hours to cultivate that a little bit. Then you can use MALDI-TOF. And then you would wait 24 hours to another 72 hours and do the antimicrobial resistance measurements. Because during this time, once you have identified the overall species of the microbe, you can use certain antimicrobials that are known to work for that species. So for instance, you know there might be some antimicrobials around that work for an E. coli infection. And then you put all of those on different plates, and you literally try out which one works best. And after up to 3 days, you take a look at the resulting plates, and if there is and you check manually essentially whether the microbe has been destroyed or not, whether it's resistant or not. And this then results in your treatment options. So based on these measurements, you do the treatment of the patients and you prescribe a certain antimicrobial regimen. So for instance, you could say, okay, now we give you ciprofloxacin, or I don't know, cefepime, and whatever those antimicrobials are called. By the way, there's not a lot of them. I thought that there would be thousands of them, but apparently in clinical practice you can count them. They're in the lower 100s, or maybe even less than 100 that are being actually used in clinical practice. And our idea is to shorten this process now a little bit by branching off here. So since we already have the MALDI-TOF data, instead of waiting for up to 3 days before doing a proper treatment based on the plate measurements that you have, we thought that it might be a good idea to use the MALDI-TOF spectrum in order to do this prediction of the antimicrobial resistance, and then already use that to skip these steps here, to skip the waiting time and prescribe a treatment directly. And in the following, I'll show you to what extent that works and what we can do with this. I mean, it's of course clear, if we would be 100% accurate here and we could predict everything from MALDI-TOF, then we would have solved this, and there would be no need to wait in addition to having the other samples collected. But unfortunately, we're not there yet. To understand why we're not there yet, you first have to understand a little bit of the complexities of the data sets, because if you use those spectra in practice, there's a bunch of steps you have to do beforehand before being able to make any sort of practical predictions from that. In the state-of-the-art package called MALDI-Quant, which is a versatile R package for the analysis of mass spectrometry data, quoting directly from the authors, it's a really great package. I'm really grateful, I need to stress this here, really grateful for the open source community for handling this and for taking so much time out of their other research operations and making this package available. It's really, really helpful. Do check it out, please. But as you can see, it has a bunch of steps before being able to use the data in practice, such as variance stabilization, smoothing, baseline removal, which is the baseline of the measurements itself, some intensity calibrations, so adjusting the heights of the peaks, and some intensity trimming, so cutting off peaks that are too large or too implausibly large, I should say. The other pipeline that we wanted to try out is we wanted to treat the spectrum as a function itself and use the so-called prominence or persistence of critical points as a proxy for the heights of a peak. We want to do this primarily because this baseline on this pre-processing pipeline on the left-hand side has a bunch of parameters that you have to pick. And the overall question of to what extent these parameters influence your outcome has not really been answered in a theoretical or in a foundational way. So maybe that would be a good way forward towards new outcomes of this research, figuring out to what extent the results are contingent on the parameter choices. But so one of the things that we wanted to try out is we wanted to see whether we can simplify this thing. Now, as a small digression here, what are critical points of a function? Well, you might remember those from your high school days. So the critical points are the points where you have either a maximum or a minimum or a saddle point in higher dimensions. I don't want to go into the specifics here, but if you think of your function as kind of a mountain range, then the prominence of a critical point is dependent on its environment. So for instance, the prominence of this critical point is definitely just its height, because if you want to climb that mountain, that is the height that you have to accomplish in order to make it to the top. But if you look at the second mountain, if you're already at the first mountain peak here and you want to reach that one now, the overall prominence is actually just the difference between this peak here and the contour line from which you can reach a higher peak. So that is actually, I didn't make this up, this is actual mountaineering terminology, because the mountaineers figured out that if you have a few peaks that are very, very close to each other, and they all have virtually more or less the same height, it's a little bit it would be a little bit weird and even misleading to mountaineers to say that, well, no, we have 3 peaks and we scaled all of them and they have heights of 3,500, 3,400, and 3,300 respectively, right? So if you can reach all of them from within the vicinity, then climbing from this peak here to this peak here is a significantly or substantially smaller endeavor than climbing from 0 to this peak here. And that's the idea that underlies the concept of prominence. As I said, I don't want to go too much into the details here, but as a pointer towards further explorations, if you're so inclined, you can think of prominence also under the name of topological persistence, which is a concept from topological data analysis. And Felix and Michael, two of my collaborators, and we wrote I hope, nice survey on this, in particular with respect to using it for machine learning. We call this a survey of topological machine learning methods. Not the most creative title, but I think it encapsulates what is going on here. It's an open access publication, so do check it out if you want to see what is going on. Now, it would be remiss of me to not say that this can, of course, also be generalized, because typically our functions are not just 1D as the spectra, but they will also be higher dimensional. This will be a useful tool later on for the MRI data set, so you can forget about it for now. But I just want to tell you that there is kind of a well-defined mapping from these higher dimensional functions over some kind of domain to a topological descriptor that is known as a persistence diagram. The exact specifics of this mapping are not important for this talk, but I want to stress that this descriptor, in a sense, contains all the critical points of the function at various dimensions. So it can sort of be seen as a signature of the function that you put in there. And under certain assumptions, it's even possible to go back from this diagram to this function here. Of course, there are certain things, there are certain transformations that this diagram cannot capture. So for instance, you can change your domain, you can translate it and rotate it, but that is by design. You don't want to be susceptible to these types of changes. So bottom line for this slide is that we have a mapping from high dimensional functions to low dimensional topological descriptors of their variability in terms of critical points. And this is what we will be using in the subsequent slides. So as a brief example of this topological pre-processing, we can take this signal here to be a one-dimensional function, and we can map every point to its relative prominence, because as I said earlier on, the prominence depends on the peaks in the neighborhood. So it's quite possible that you will find that a lot of the peaks get already mapped to 0. And as you can see here, with the new spectrum that we obtain after pre-processing, that is the case for a lot of the peaks here. So it already removes this baseline, it does some kind of stabilization for us, and it results in a very nicely neat looking spectrum. The only downside we have is that we are kind of transforming the domain alongside with that. So technically speaking, this is not an intensity anymore, but it's something else. It's more like a prominence, but we can ignore that for the time being, and we can continue nonetheless with these types of calculations and just pretend that it's some kind of special pre-processing that we're doing here. Now, how do we use such spectra for machine learning tasks? The slide that you've been waiting for, I hope. The most straightforward way is to just bin those spectra into fixed size feature vectors. So we choose some bin size, maybe 20 Dalton or 5 Dalton or whatever you prefer. And then we bin each spectrum, we obtain feature vectors of the same dimensionality, and we just put them into a neural network. The other representation that is equally valid, but a little bit more complicated, would be to use a sparse representation of the spectrum based on tuples. So if we have, for instance, a certain intensity threshold, then we could say, okay, at these positions, we find a peak that is above a certain intensity threshold, and we just feed these positions alongside with the intensity values to a neural network. This latter part, in case you're familiar with that terminology, the latter part is a little bit reminiscent of a deep sets approach, wherein you treat your observations more as unordered sets, as opposed to the first approach with the bin size, where you get equal sized feature vectors. both have their advantages, both have their disadvantages. Depends a little bit on what you want to accomplish. You will actually find that in this talk, we'll be using both, but I'll point out which one we used for each of these applications. Now, the first one that we used is the PIKE, the peak information kernel. This uses the second approach. So it uses the sparse information gained from tuples and from intensities and peaks. I don't want to go into the details too much here, but you can think of this as follows. We have two spectra S and S prime, and we have their respective mass over charge values, xy and xy prime, and the intensities lambda I and lambda I prime. And then we calculate this, well, complicated looking exponentially weighted expression. The important thing to disentangle these equations here is to see that we use the squared distance between the x values essentially and the product of the intensities. The idea being that if you have a large peak and a small peak, they should kind of cancel each other out. And then we have some weighting factor, but we only need this because it's based on the solution of a differential equation. And without that weighting factor, this solution would be incorrect. So we want to be mathematically correct, but it would also work without that, more or less. The properties of this kernel are very nice from a theoretical point of view, but also from a practical point of view. Namely, it's a infinitely differentiable function. It's smooth also, and it has a closed form gradient expression. This last part is particularly nice because it means that we can train any type of model that makes use of a gradient. As I said, the interactions between peaks are somehow captured because you can see that this depends on a product. So we go over all the peaks in all the spectra, and we use a similarity measure based on peak distances. Now, the final last straw that we liked about this approach is that we have a single parameter that controls the smoothing. So this t parameter is a real number, and setting this makes it possible to smooth the spectrum accordingly and yield a kind of multi-scale view of the spectrum at different resolutions. We'll see how that looks in practice here. So for no smoothing, we just have the individual tuples. So as I said, x positions and some y positions, so nothing going on here. I've sorted them, of course, accordingly to their position. Then we can add some smoothing, and it suddenly becomes a function. I can add some more smoothing, some more smoothing, some more smoothing. And you can see that the peaks get increasingly pulled down because they are relatively isolated. So it's a very nice way of smoothing down an individual spectrum and comparing spectra at the proper resolution. And of course, if you continue this process, at some point you will just have a flat line. But notice that the variation is still quite large. It's just that for the sake of this visualization, I did not change the axis here. So all of them live in the same coordinate system for this visualization. But if you would now zoom in, you would still see quite some variability. Now, how do we use that? We use it to predict the antimicrobial resistance for a small test data set. I mean, relatively small test data set. So it's about 1,000 samples from specific species and from specific antibiotics. I don't want to go into too many details here, but you can see that we picked 3 species that have been declared priority pathogens from the World Health Organization. So these 3 species alone are responsible for quite a few infections in the clinical practice. So if we have some way to target those specifically, that would already be nice. You can also see that we picked a bunch of different antimicrobials. Notice also that some of them are being used for different species. So for instance, ciprofloxacin is being used for all 3 species here with, of course, varying successes. The last point I want to make for this slide is that if you take a look at the percentage of resistant pathogens or resistant samples, you will find that this varies a little bit. So it's definitely not a very balanced data set, which also means that we have to account for this during training. But you can also see that if we give penicillin, for instance, you will find that almost 70% or more than 70% of all the species are resistant towards this. So you can easily see that normally it's the resistant class is the minority class, but for some pathogens it might actually flip. Now I should say that this is taken from our publication Topological and Kernel-Based Microbial Phenotype Prediction for Maldive mass spectra, where the idea was that we wanted to see how far we can push this sparse kernel approach with the peak information kernel that I showed you before. Now, I should say that in this case, it worked spectacularly well. So we trained a Gaussian process classifier. The Gaussian process classifier represents the sample, or represents the prediction as kind of a multivariate normal distribution, and it allows for integrating a smooth kernel function such as our kernel. And in particular, and this is now where everything comes together, it allows finding the best T parameter, the best smoothing parameter based on the training data using some kind of optimization scheme, as you would do for a neural network. So that is why the Gaussian process classifier was a really good choice here. We compared it to a Gaussian process classifier with another kernel, so with another similarity measure. Here, this is a so-called RBF kernel, a radial basis function kernel, which is essentially a polynomial assessment of differences of the function. And as our baseline comparison partner, we used a logistic regression, which is what people have been using in this field before. So you can see that the Gaussian process classifier with the new kernel works really well. It improves over the baseline, sometimes quite substantially so. In other cases, the gains are more modest. So it was a kind of nice test balloon for this sort of approach. In particular, what we liked about this early on was that we have this sparse processing. So we are looking only at sparse data sets. And moreover, we can get a built-in confidence analysis from the classifier, because the Gaussian process can give us, by virtue of being a probabilistic method, it can give us access to the likelihood of specific samples occurring. And so we can easily later on calibrate the classifier and make sure that if the likelihood is too low, we could reject a prediction. This is very important for the clinical practice, because if your classifier is confronted with samples that it hasn't been trained on, you should be able to detect this early on and to reject a prediction if possible. Now that all sounds nice and well, of course, but we did not continue with this approach, honestly, because we found that it has some insufficient scalability to larger data sets. So of course, having sparse representations is kind of nice, but in the end, you're also throwing away a lot of the information about the spectrum. So essentially the choice was, should we keep on trying to make this approach a little bit better? There's not a lot of options here, because it scales quadratically with the number of peaks or with the number of tuples that you put into it. Or should we try to move towards something that works with a full spectrum and that doesn't throw away any information? And we opted for the latter, at least for now, but of course we will keep this approach in the back of our minds. And maybe there are situations in which this is more applicable. For instance, when you have a high throughput mass spectrometry data set, which might not be what you have in the clinic, but who knows, maybe there are other application areas where this could shine, essentially. Now, I call this slide bigger and better because it's kind of the natural progression of what we started out doing with the first publication. Essentially, we started collecting a large number of mass spectra with a focus on the priority pathogens, of course, but also with a focus on having clean data. And this, I should stress, is a massive effort from a lot of people. In particular, Carolyn Weiss, the first author of this publication, had really the bunch of the work here, making sure that all the pipelining worked and that the data transformation was consistent and so on and so forth. So we are also really grateful to our clinical partners for making this data set available. And we even made it available now to the general public. So there's a DOI link. I think I'll also share the slides later on anyway. So please do check that out. It's really a nice data set. It contains 300,000 mass spectra with almost 800,000 antimicrobial resistance labels. So every mass spectrum has a bunch of different labels attached where antimicrobial resistance was assessed different antibiotics. In total, there's 800 different species of bacterial and fungal pathogens. And to put the cherry on the top here, it's from 4 different diagnostic laboratories. So, and these are, by the way, and these are disentangled in the data. So you can easily download one lab and train on that and then see whether your data generalizes. We'll see that it's not as easy as I make it sound. The publication that this was now described in focuses primarily on scalability. So figuring out how to use some standard classifiers that work quickly based on just 6,000 dimensional feature vectors, which we got from binning the spectra with some kind of predefined M over Z range. There's also other experiments in this paper, of course, but I want to stress that this is just the first step towards making use of this data set. And I really hope that there will be more publications coming out and more analyses going forward in time that make use of this treasure trove of data. Now, to show you some results which made us relatively hopeful in the beginning, we find that that a logistic regression, which is the solid line, and the light GBM classifier, which is the dashed line, they perform relatively well on these ROC curves, as well as on the precision recall curves. But you can already see that given some different antibiotics, the performance can differ quite drastically. So the usual ROC curves that you're looking at, they are a little bit misleading since the overall class imbalance might change. And so what you should be looking at if you want to really critique the classifier and find out what is going on, you should look at these precision recall curves here. And there you can see a little bit of a different behavior. It's also not, I would say that from this, it seems that that logistic regression is always performing worse. So the dashed line is always below the solid line. The solid line is always below the dashed line, so below the light GBM classifier. But you can see that for certain combinations, this is not true anymore. And of course, you can imagine that we have many of such plots for different pathogens. So this is for E. coli, and we have several other ones for other combinations. And there's no clear picture. There's no clear winner emerging, which is a good message, I would say, because it means that even a standard logistic regression, if properly trained and calibrated, can contribute something here. And it might be preferable for certain applications because it might be easier to train or it might scale more nicely. We also have some neural network experiments and results in this paper, but I should stress that those are rather preliminary. Also one, well, not so much success story here. None of these curves reach the point of clinical relevance or clinical applicability according to, I think it's FDA guidelines. So to really turn this into something that could be used in clinical practice, we need either more data or a better way to improve these performance values here. However, I should also stress that it's a very complicated task because MALDI-TOF was not meant to capture these mechanisms for resistance prediction anyway. So the fact that we are getting anything above a random baseline is already very nice and encouraging, I would say. And who knows, maybe this could be used as an assistive tool as well. Now, before moving to the second part of the talk briefly, I want to show you some nuggets of things that we encountered later on and that still plague us to this day. And we're looking forward to getting better answers from other researchers here. The first thing I want to show is what happens if we move to other sites. So this is a classical ROC heat map. So every cell shows you the area under the receiver operating characteristic curve. And the cells indicate where you trained your classifier. The rows are the training site, the columns are the testing site. So all the diagonals are the values where you train on one side and also test on this side. So of course, with the different training test splits, so you're not double dipping into the data. I just want to make this very clear. But what you would now expect is as you move to a different site, you would probably expect that the performance drops, but you might want to hope that the performance drop is not too large. However, you can see here that there's some quite some weird things going on. So first of all, going from site A to site B, performance actually increases, which might mean that they are looking at a different collection of pathogens that are maybe easier to predict, because those hospitals, they tackle different cases. So it might very well be the case that the easiest infections go to one hospital and the harder ones go to the university hospital, vice versa. But overall, there's not a clear picture. In particular, it's tough to predict the performance in advance. So for instance, going from site D to site B, you have almost the identical performance, but going from site B to site D, you have a drop in performance. So there's clearly something complicated is going on. I don't want to say that it's wrong. I mean, these numbers make sense, and they just indicate that our classifier doesn't generalize as well as it could or as it should for clinical practice. We zoomed into this also and found that there are quite a few confounding effects at work here. So for instance, if we look at the spectra from different sites, for one pathogen only here, for instance, we find that the B site clearly stands out in a very simple plot. I mean, of course, those annotations have been done after the fact, but you can find that all the green dots are coming from one hospital, and all the violet and orange dots are coming from the same hospital, albeit with different Malditov machine types. So the fact that these single spectra are situated at the boundary of the embedding might already give you some head scratching, because there's clearly some kind of confounder at work here if we are able to disentangle that. We also trained a classifier to predict the site a spectrum came from, and that also worked relatively well, which underscores the problem that we're dealing with here. I should stress that these confounding effects, there are no trivial confounders in there. So it's not like we missed something very obvious. I would hope so. I think that I think the way this works is that every university follows a kind of protocol to make these predictions, and notionally they all follow the same protocol, but still we see these deviations. So clearly there's something that we need to investigate further. It might be related to the microbial landscape, might also be related to the way the machines are being administered or are being managed in practice. But we can see that there's clearly some work to be done here. Now, some lessons from this project. I'm happy to report that even relatively simple models, so like logistic regression, a light GBM, or a multi-layer perceptron, already exhibit useful performance. The calibration of such classifiers is absolutely crucial in order to support the rejection of samples that we don't want. And moreover, in the draft and in the publications, we also show that we can use feature importance values of a trained classifier in order to highlight some interesting peaks in the spectrum, kind of like an attention that tells us where the classifier was looking. But of course, there are challenges. There's this whole domain adaptation or domain transfer challenge that I just mentioned, but also, and maybe this is related or linked to the first point, we need to extract features that generalize still over time, because the antimicrobial landscape is constantly changing and shifting. So strains might appear, strains might disappear. We all saw that during the past years, unfortunately. And so having a representation that works well there is absolutely crucial. Now, with this, I would briefly talk about the second part, if I still have some time. How is that possible still, Daniel, or should we?
I mean, depending on how short it is, maybe a few minutes is fine within, so that we still have some time for questions.
Yes, yeah, I'd love to take some questions. I just want to highlight some nuggets here, because I really like this. This is something that is very dear to my heart, because it's a very new technique that we're trying out, and we're trying to push this a little bit towards fMRI data analysis. So it has nothing to do with antimicrobial resistance, but I want to briefly show you why this is important. So you get these nice functional magnetic resonance imaging data sets. There's a cut through the brain. The resolution is not as nice as it could be, but you can see that there's some kind of changes going on over time, and capturing this is still a very challenging task in practice. And what we did in our approach here is we used we considered this bold activation function, so this blood oxygen level dependent activation function that researchers are measuring. We consider this to be a time varying function on a manifold, so similar to what I showed you earlier with these mountains. And then we calculated the topological features of this function on the manifold, which makes it possible to obtain stable topological summaries at different resolutions. And the nice thing, the main advantage of this approach is that we can work directly on the raw data. So there are no auxiliary representations necessary for processing these MRI datasets. We can almost take them as they come out of the machine. And in particular, we don't require an atlas. So neurologists or neuroscientists typically use an atlas to decide which regions to look at and how to segment the resulting representation. We don't need to do anything of this sort. So in total, there are fewer modeling choices, which is always a good thing to have in terms of Occam's razor. So having a stable representation that yields some information already is a very beneficial thing here. I don't want to go into the details here because, yeah, I just want to sort of sketch out what we're doing. Essentially, we're going from a stack of fMRI data sets to a topology-based representation that tells us the topological activity with respect to this data set. And we can make use of these topological descriptors in a variety of tasks. The most interesting one would be an age prediction task that is kind of a baseline smoke test for neuroscientists in the community, apparently. So they take all the participants that are not yet adults of this study. So by the way, no children were harmed in this study. They were just watching a movie, like literally they were shown a very cute Pixar movie, which was really soothing, and then their brain activity was being measured. And so one thing that we tried out as a smoke test here is to see whether we can predict the age of the participant or whether that correlates with overall topological activity. And the result, I want to direct you to the last row of this table. The result is that it does, and it does that quite well. So we get a correlation coefficient of 0.61, as opposed to the best neuroscience method that was available at the moment, which was 0.44. It's an SRM based model, which also has some modeling choices in there. So this proved very, very hopeful for the future, and we hope to extend this approach to a bigger range of tasks in neuroscience, because, well, for now we've really just scratched the surface of this, and we really just have a very basic task and can do some quantitative analysis here or some qualitative analyses here, which tells us something about how adults and children are perceiving the world. But I'm going to stop here now and rather want to thank all the collaborators of these various projects. So there's quite a few universities involved here. In particular, given this antimicrobial resistance prediction task, quite a bunch of people from Basel University and of course from ETH. Also for this fMRI dataset, this was a collaboration between Yale, Université de Montréal, and ETH Zurich. Funding was graciously received from the Swiss National Science Foundation. I'm really grateful about this. And as I already said in the beginning of the antimicrobial resistance part, special thank you to all open source contributors for making this science possible and for making science possible in general. So I'm going to stop here. I had my say, and I'm looking forward to a lot of exciting questions now.